High-content screening systems allow for a variety of morphological and cytological analysis to be performed simultaneously on many samples in parallel. In this video, neuron astrocyte co-cultures are prepared and seeded onto 96 well plates where they are treated with a neurotoxic agent and subjected to various neurotoxicity stains. Plates are loaded into the GE in-cell analyzer and the investigator software is used for analysis. From these plates, readouts for neuro and glial toxicity such as neurite length, neuron count, GFAP expression, and astrocyte area can be obtained.
Hi, I'm Andrew Ball, Senior Scientist at Millipore Corporation in Temecula, California. Today, Janet Anderl, Research Scientist for High Content Analysis, will demonstrate methods for high content screening of neurotoxicity using neuronal and astrocyte co-cultures. This technique employs reagents and protocols from the Neurotox-3 assay developed here at Millipore to screen for neurite outgrowth, neuronal and astrocyte development, and neuronal and astrocyte toxicity. I hope you enjoy the following sequences.
High-content screening for neurotoxicity begins by seeding neuron astrocyte co-cultures in 96 well plates. Once co-cultures have grown to approximately 70 to 80% confluency, detach them using trypsinization. Cells should be counted and adjusted to the appropriate density for the assay before they are seeding in the plates in 90 microliters of growth medium. Cells should be